Neoplasia. So the purpose of this talk is to give you sort of a basic foundation of what neoplasia is. And so what I've done, I've set up a couple of different ways. One, we're going to go through some of the general properties of neoplasia. Then we're going to visually show you what these neoplasms look like. And specifically, we're going to take a specific part of your body, colon, and then show you what a benign neoplasm is and a couple of malignant neoplasms. Then we're going to get into the biological properties of neoplasia and some of the causes of neoplasia. We're going to go through chemical carcinogenesis. It's a very interesting model that's been developed, and you'll see that a lot of the concepts and models that developed about chemical carcinogenesis came from U of T, which is kind of neat. And then we're going to get into cancer stem cells, and that's a pretty hot topic nowadays. But I do want to emphasize that I'm going to throw a lot of information at you, and basically I make no apologies. So let's get on with it. Neoplasia. So what is neoplasia? And throughout this talk, you're going to hear me say neoplasia, neoplasms, etc., etc. And I will not go and call them tumors. You might hear the odd tumor name, but I'm going to try my best to essentially not use that word. Why am I doing this? Well, what is a tumor? Most people refer to a tumor as a cancer. Is that the correct definition? No, it's not, but we all use it. When we look at the word tumor, and look at the meaning of the word tumor, and I alluded to this earlier, tumor, by definition, is any localized swelling. So that makes sense for a tumor. Basically, a neoplasm. You have a local swelling, a local area that has an increase in mass. And that's what a tumor is, right? Well, not really, because a tumor is any localized. It can be any localized increase in mass, and that could be from an acute inflammatory reaction, which tumor, by definition, is one of the cardinal signs, and we'll talk about that in a third year course. The proper terminology is neoplasia, and when we refer to basically the mass, it's a neoplasm. So you will still hear the word tumor from everyone, and unfortunately the literature is full of that in terms of the word tumor, and by convention now, that is what has taken the place of neoplasia. Anyways, just so you know, I'm going to try and avoid that word as much as I can. Not that I don't like the word tumor, it's just the meaning of it. So basically, neoplasia is literally defined as new growth. Neo, meaning new, and plasia is referring to the actual cell. Now, what exactly is it? Well, it's cell proliferation and growth in the absence of any environmental perturbation. You're losing control of that specific cell. That's the whole concept behind neoplasia. Thus, this term neoplasia refers basically to this autonomous growth. And the word neoplasm is the resultant abnormal mass. So when you refer to the tumor, it's the neoplasm. And there's many, many, many factors that contribute to neoplasia formation. And we're talking basically irradiation, chemical carcinogens, and there's plenty of those in the environment. And so we're going to emphasize quite a bit in terms of chemical carcinogenesis. And before I proceed, I just want to let you know there's an article on Quercus that deals with this and deals with the models that we're going to be talking about later on. So a lot of that work was done here at U of T with Emmy Farber and Dr. Sarma in laboratory medicine pathobiology. Very interesting read. Look at the date of it. Back in the early 80s, 1983, 84, and around there. And that model really has withstood the test of time with many aspects of it. We're going to talk about those as we progress through. Certain viruses can actually cause cancer as well. Plus, there's many, many other causes that we just don't have time to deal with. So when we're looking at neoplasm, specifically, we can divide their behavior into two major groups. One is called benign, and one is called malignant. So benign is a mass that's well-defined, and cell proliferation and growth is very local. Versus malignant, which the mass is very often poorly defined, and the cells can grow into the surrounding area, into the surrounding tissue, if you would, and you can actually destroy it. You can cause tissue injury within that area. We call that local invasion. In addition, these malignant neoplasms have the ability to spread to distant sites. We call this metastasis. And the resulting neoplasm, the secondary neoplasm, is the metastasis. That's not to say that a benign neoplasm is not lethal. It can be. So if you have a benign neoplasm within the brain, it can be quite lethal. Why? Because it's going to grow. And if you look at the structure of your skull, you have a finite volume within that area. So if it continues to grow, it's going to put a lot of pressure, and it can be lethal. It can continue to grow. Also, many malignant neoplasms are what we call ectopic. And that means that they secrete a variety of different cytokines and hormones and things like that. And usually when it does this, this leads to the clinical symptoms. So, when we're looking at the characteristics of a neoplasm, you have this abnormal cell proliferation. That's a foregone conclusion. So it's very abnormal. And we'll have a look at that in a short period of time. In addition, another property that's really important is the degree of differentiation, or what we call essentially the maturation state of these cells. I prefer using the degree of differentiation. I'll explain why in a minute. So when we're looking at normal tissue, these cells are what we call fully mature, or they're highly differentiated. And stem cells are often described as undifferentiated cells, which makes sense. Now, when we're looking at neoplastic cells, they're going to basically span the entire gauntlet of that. They're going to have variable degrees of differentiation. And most of the time, they don't achieve this highly differentiated state, and that includes benign. So when we're looking at benign masses, then we often define benign neoplasms as well differentiated. They're not fully mature. They don't look like the normal tissue. Well, they sort of do. I'm going to show you evidence of that. They do look sort of normal-ish, but they're still a neoplasm. They're not responsive to growth signals. That's the problem. And we'll deal with that later on as well. When we're dealing with malignant neoplasms, wow, this one really spans that spectrum. You can have malignant neoplasms that are well differentiated, and others that are what we call poorly differentiated. And the name of the game here is to, when we're looking at these sections, these histology images of neoplasms, we want to see and find common characteristics that we see with the normal tissue. And that's what we try and do. And we're going to do that as an exercise together. So here we have what we call normal colon. This is a very small snapshot of your colon. And let me show you some of these cells. So here is what we call a crypt. And this is a very simple type of gland, basically. And it looks like a test tube, right? So we call this a simple tubular gland. And so basically because it's tubular in terms of its shape. Within this test tube, we have thousands upon thousands upon thousands of cells that make up this specific type of gland. And you'll see the predominant one is this ovalish shape, opaque looking cell. These are what we call goblet cells. And they secrete mucus to keep this entire colon, if you would, entirely moist. That's the whole procedure there. You want to keep this lubricated as much as we can in order to get that fecal matter traveling along efficiently. There are many other cells found within this test tube, if you would. and we'll go through those in the third year course. One of them is called the surface absorption cell, and that cell does what it says. It absorbs. It absorbs excess water and electrolytes and everything else and brings them back into your body. In addition, if you look in here, what do you find? What did we cover a few weeks ago? You got it. All these dark cells, diffuse lymphoid tissue. You got it. Perfect. And then we have a structure down here that basically is made up of smooth muscle cells, and then we have connective tissue below. So what we want to do is take a small section of this area here, and what we're going to do is show you a control and then 3 neoplasms, different neoplasms. One will be a benign, one will be a well-differentiated malignant neoplasm, and one will be, you got it, a poorly differentiated malignant neoplasm. Now you have to remember, all of these neoplasms are from the same region. So we're talking down in this area here, okay? Now as soon as I turn to it, you should be able to identify what's what, just based on visual clues, and we'll go through these visual clues very shortly. Here it is. Very obvious of which one is benign, which one's malignant. Very obvious. So remember, what we want to do is look at the basic control and compare the other ones to the control. And remember, all of these neoplasms are from the same area here. So this is where they would originate from. So this is the control. This is normal colon. You can see the goblet cells. You can see another cell type in behind. This is the columnar-ish type of cell. And basically, when you're going down the colon, it's really full of goblet cells. There's so many of them, it's not even funny. But the other cells are still there. They're just squeezed in between these goblet cells. But they're huge numbers of goblet cells. As you go down, essentially from the large and small intestine, all the way down through the rectoanal junction, the number of goblet cells per unit area increases along the entire length. So back to this story. So this is what the normal tissue here looks like. Let's take a look at this image here. What do you look for? What do you see? What you should be looking for are common features that you see that look similar to the control, similar to the normal region here. They're not going to look the same. It's obvious. This really doesn't look very normal, and it's not supposed to. Okay? What we're looking for are common characteristics that allows us to essentially tell where this neoplasm originated from. So what do you see here? Look for the common characteristics. So if common characteristics include, and we're looking over here, we're looking to see whether or not, because the colon predominates in terms of goblet cells, we want to see, do any of these cells actually have mucus production within them? Are they columnar in terms of their shape? What does the nucleus look like? Is there something strange about the nucleus? Is it elongated? Is it bigger than normal? Does it have a very distinct nucleolus? Does it look normal? Does that nucleolus look very normal? Is there evidence of what we call mitotic figures? Why? Because, well, it's the neoplasm, right? These neoplasms grow. They don't know when to shut off because they no longer have basically control over the growth process. That's the problem. So look for these common characteristics. Do they form sheets? Do they form cords? Do they form nodules? Or do they basically none of this? And we'll have a look at that one a little bit later. But for now, have a look at this one here. What do you notice? Well, do you see any evidence of mucus being produced? You do, right in here. All of this is mucus being produced. Perfect. Okay. What about the shape of the nucleus? Yeah, they don't look normal. They're quite much larger than what you would expect. You do see the odd nucleolus, and it doesn't look too weird. So we keep that as a feature as well. But you can see there's more crowding down in here. Much more crowding. So that's another indication. Here you don't see that crowding associated with this type of neoplasm. What about the shape of the actual cell itself? It's hard to see, but here in the goblet cell, we can actually see that it's very ovalish in terms of its shape. Here it's very tubular, isn't it? It goes straight up and straight down. So what do you think this is? If you were to give it a name, what would you call it? Yes, if you're thinking benign neoplasm, you are correct. Good. Now, the actual name we give this is what we call a tubular adenoma. Tubular because it looks tubular, and adenoma because it really, when you're looking at the word adeno, that refers to glandular origin. So here we have a glandular origin because we have mucus being produced, and because it's benign, we give it the ending oma. So this is what we call a tubular adenoma. It does not look normal, okay? But there's enough evidence to say, yeah, we've got production. It's pretty much well differentiated from that perspective. And so this is an example of a benign neoplasm. Let's go to this one here. This one looks completely different than what we just saw. What do you think is happening here? Do you see any common characteristics? Do you see any evidence of mucogen being produced? What about mitotic figures? Do you see any of those? What about the shapes of the cells? Shape of the nucleus? What do you see there? Well, let's have a look. Yeah, I see a little bit of evidence of mucogen being produced right here. You can actually see the outline of some of these cells. They're still columnar-ish in terms of their shape, but the nucleus, definitely not normal looking, very enlarged. You can see that. Many have multiple nucleoli, but some of them look very abnormal. So here's one with a little tangent coming out of it. Here's another one that looks weird too. Another one that's very hyperchromatic. That's another characteristic sometimes you see within neoplasms. Another one here. In fact, this right here and this right here are what we call mitotic figures. This is essentially an active stage of mitosis frozen in time. And sometimes these cells, when they're proliferating, will actually undergo very abnormal mitosis. And as a result, they'll be fixed at this stage permanently. They will not be able to proceed. Because in order for you to grow, in terms of the cell, you have to undergo complete mitosis. And if you have an abnormal stage of mitosis, chances of completing mitosis is poor. As a result, you're going to be stuck. And the histology image will catch you right there. So here we have evidence of that mitotic figure. Now, as you go from what we call the benign through the malignant category, the number of mitotic figures, abnormal mitotic figures, increases. And we'll see that in a second. So back to this one here. What about the shapes of the cells? We saw that they were columnar in terms of their shape. Okay, so this does show similar characteristics to this area here. You know they're definitely not normal, that's for sure. So we would call this one, based on the criteria that we just had, we would call this a malignant neoplasm of the colon. In fact, I would call this one a well-differentiated malignant neoplasm of the colon. Okay? What about this one here? All bets are off, right? This one does not look at all like this area here. Even though it's from the same area, no similarities at all. You can see the cells are larger, the nucleus is very, very different, very abnormal looking. You can see within this area here, nucleoli are very abnormal looking. We have mitotic figures in quite a number of different areas. In fact, we see more mitotic figures than we do within the well-differentiated malignant neoplasm. Here we have a few. Here we have many. You can see them here, 1, 2, 3, probably getting into this one here as well. And this one looks abnormal too. So there's many telltale signs that shows that this is very, very abnormal. The cells, large in terms of their size. Sometimes the nucleus can be quite large. Here they're very different looking. Sometimes they're very dark in terms of their staining. But there's a very abnormal scattering or invasion within this localized area. And this invasion could actually take on different forms as well. You can actually spread them throughout. But in terms of having minimal characteristics, what would you call this one? Definitely, this one is a malignant neoplasm, and it's a poorly differentiated malignant neoplasm. Very much so. Here's another one. This is called a leiomyoma. And by definition, it is of the smooth muscle cells within your body. You can see it's very nodular in its appearance. It's basically almost very spherical in terms of its shape. And this is the area that we're looking at is what we call the myometrium. And it has a very thick wall of smooth muscle cells within this area. But occasionally you get what we call the leiomyomas. And these are benign neoplasms of the myometrium. And it's semi-common also. So let's have a closer look over here. As you can see, this is the actual neoplasm. It's benign. And this is the normal tissue found within the myometrium. It does look similar-ish. But because this is a mass that's growing, and because we can actually see some of the similarities between these smooth muscle cells and these smooth muscle cells, we know that this is a benign neoplasm of the myometrium within this area. In fact, if you look closer, you can actually see how it's squeezing in the smooth muscle cells within this area. And we call this a pseudocapsule. Pseudo meaning false, capsule as it looks like it's being encapsulated, but it really isn't. It's just a pseudocapsule because it's pushing in these cells and it's giving the appearance that it's encapsulated, but it's not. So in this area here, this is all benign neoplasm, and it grows. But it doesn't grow ultimately fast, and most people live not knowing that they have leiomyomas within them. Now we're going to look at breast cancer, and I'm going to show you a normal mammary gland. And what we're looking at here, this is where these are the ducts that contain the glands, and the glands are the ones that produce basically the milk. And mixed in amongst these lobules, if you would, is connective tissue. So all of this area here is connective tissue, and you have a bit of adipose tissue mixed in within the lobules of the milk-producing glands. So this is the normal structure. And when we look at breast cancer itself, you see huge differences, and this is of the same area, where these glands, once, when they're forming these sort of tubular systems, actually start to grow in cords and almost nodular appearance, but very much like cords. So all of these, they're outgrowing like this. And we have a lot of collagen in between that we can see within the lighter pink. So all the darker areas, these are all the cords of those ducts that are essentially growing. And they grow and grow and grow. Here's a close-up image of the same effect. So we have connective tissue in between, the lighter pink, and then these cords are all of these. This was those round, circular, basically duct system that produces the milk and transports the milk all the way through. But here, within breast cancer, they form these long growth cones, if you would, all the way down. So these are the cords that make up this type of cancer. And all these dark cells that you see is a mixture of cells that come off and start to invade localized areas, plus lymphocytes. So there's an immunology effect that we're having within this type of mass. And this is a malignant neoplasm of the mammary tissue. Let's get into lung carcinomas. These are really interesting. And the reason why we chose lung carcinomas is we've done a lot in terms of the lung. And so this will be our representative model system that we're going to look. You know what normal lungs looks like. Now we're going to show you what benign and especially the malignant neoplasms, we're going to concentrate on the malignant neoplasms of the lung. So when we're looking at lung carcinomas, and we'll define what this is very shortly. Benign lung neoplasms are very rare, usually extremely rare, and if you'd have it, you'd consider yourself lucky, because malignant neoplasms are the predominant feature of lung carcinomas. And that's where the term comes from. Carcinoma refers to a malignant neoplasm of epithelial origin. In other words, the cells that line your essentially trachea and main bronchi, especially the bronchi, are basically where these neoplasms would develop. So the majority of these lung neoplasms are carcinomas. They're malignant and epithelial origin. And they arise in the bronchi, and hence we give them the term bronchiogenic carcinomas. And there's usually four major types of these. What we call the squamous cell carcinomas, which we'll have a look at. Adenocarcinomas, we'll have a look at that. Small cell carcinoma, which we won't have a look at. Really, it's a different type of cancer. And it's actually one that really has a very poor prognosis. And the other name that they give it is oat cell carcinoma because of the way they look. Very oval in terms of their shape, and they actually look like oat cells. The cereal that you eat for breakfast, oat cells. That's what they look like. Another one is called bronchoalveolar cell carcinoma. This is, although it's rare, it's actually a really bizarre type of cancer. And I'll show you exactly what that looks like when we get to it. And this gives you a hint. Crowded columnar epithelium. Very, very nasty. So you know what the normal looks like. And we've gone through this before. And we have a bronchiole here, followed by the terminal bronchiole, just to refresh you, leading into the respiratory bronchiole, which has the alveolus. So this is the first place we'll have respiration taking place, followed by the alveolar duct and ending in the alveolar sac. That's what we're looking for. And, of course, the alveolus, located here. This is the one. It's called bronchoalveolar cell carcinoma. This is where those simple squamous epithelium that line the alveolus now changes to a completely different shape with this type of cancer. It becomes very columnar, or very large and tall in terms of their shape. Now, can a person breathe with this? Can you get gas exchange taking place within these cells? The answer is no. These columnar cells are too large for gas exchange to go through them. In order for gas exchange to take place, we need those beautiful, simple, squamous cells, very, very thin. That's where gas exchange can take place very easily. When you go to a larger cell, it just doesn't happen. That's the problem. So we have invasion of what was the simple squamous cells. They're now being replaced by all these columnar cells, very columnar-like in terms of their shape. And they're taking up all that surface area. In addition, you may have noticed that some of these regions are quite thicker than your normal alveolus. And that's fibrosis happening. So we have this whole chronic inflammatory response that's happening within this area. So we can get some tissue destruction, fibroblasts coming in and laying down much, much unwanted collagen within these areas. So this is where fibrosis is taking place. All these dark cells, what do you think they are? You got it. These are all inflammatory cells. These are all lymphocytes coming into the scene as well. Very nasty cancer. Very poor prognosis in terms of how far this spreads. If it spreads too far, you're not going to be able to breathe. It's just very nasty. Let's get into what we call the squamous cell carcinomas. This is probably the more interesting lot of the neoplasms that we're going to cover. And why? Because we're going to show you two different versions. We're going to show you a well-differentiated malignant neoplasm of the lung and a poorly differentiated malignant neoplasm of the lung. And you'll see how the squamous cell carcinomas, and why we call them squamous cell carcinomas, essentially arise from what we call the main bronchus. And usually this is where we see what we call squamous cell metaplasia. Now, what exactly is that? Well, if you're a smoker, and you're continually smoking, or there's some sort of chemical irritation that's happening along the epithelium of your respiratory tract, within your trachea, within your main bronchi, what happens? Well, the irritation has an effect on what we call the respiratory epithelium. And normally that respiratory epithelium is very columnar in terms of its shape. So these cells are quite large. Some of these are ciliated, and these cells move around mucus and get rid of all the particulate and some of the pathogens, etc., etc. So that's what normal physiology is. But if these cells undergo constant irritation, chemical irritation, physical irritation, then sometimes what happens is we have a change in the actual shapes of these cells. And one of these changes, and we call this change from one epithelium to another type of epithelium, we call that metaplasia. Specifically, the shape that seems to withstand the chemical irritation are squamous cells. They're flat and they're quite strong from that perspective, and usually you get a thick layer of them. So we call this squamous cell metaplasia. And this occurs within those areas within the main bronchi especially that's undergoing constant chemical irritation. It could be from cigarette smoke, it can be from other types of chemical irritations. And this area here, what we call squamous cell metaplasia, is thought to be the origin of some of these lung cancers, specifically the squamous cell carcinomas. And these masses can actually invade local parenchyma, which is the areas around it, and it can obstruct these air passage, which is not very good. And you can also get the metastatic variety, which can actually invade the vascular system, and usually via the lymphatic system, it's going to go to the lymph nodes, and via the normal vasculature, if you get what we call tumor vascularization happening, basically it can metastasize to distant sites within your body. In addition, the degree of differentiation can vary widely with this type of tumor. That's the problem. And we can be what we call well differentiated. You actually might see features within the mass that share normal characteristics to what we call stratified squamous epithelium, which I know you haven't had histology, but one of the major places that you see this type of epithelium is on your skin or your esophagus. It's a slightly different type, but let's take the skin as the example. So within your skin, and we'll show you a picture very, very shortly, you have squamous cells, a rather thick layer of those cells, and then because your skin is what we call keratinized, you will see usually a pretty thick layer of keratin, which is an intermediate filament. And so as these cells move up the scale from the basal component, and we'll show you that in an image in a minute, as they move up, they get flatter and flatter and flatter. They eventually die, and what's remaining is keratin. That makes up the most outermost part of your skin. Interesting. Anyways, where's this leading? We just want to show you that image first, and then we're going to pick out common characteristics that may show evidence of, say, keratin being produced within the tumor, within the neoplasm, and other characteristics in terms of what these cells look like. So those are the common characteristics that we want to show you, as well as are these cells actually growing together, as in sheets or nodules or cords. Those are the other common characteristics we want to look for. If they're not, then it's very, very hard to say where that neoplasm originated from. In addition, if it's very well differentiated, you may not see many mitotic figures like we talked about earlier. Interesting. So keep those common characteristics in your little tool chest, and then we'll have a look and see what's being produced by these cells. Other things that you want to look for is evidence of keratin being produced, especially as what we call these little masses of keratin. If you have a little mass of keratin, we actually call that a keratin pearl, and that looks pretty cool. We'll show you that as an image. First, and a little more in terms of what we call the poorly differentiated variety. And the problem here is that we're not really looking at any characteristics. They're just not there. You have this big nodule that is completely full of what we call large cells. And that's the problem. They all look large in terms of their shapes, and in terms of the mass, you won't see other common characteristics that you would normally see in using skin as our example. So you wouldn't see any keratin being present. And we classify these large cells, and we can just give them a very generic type of name. We call it large cell undifferentiated carcinomas, or strictly as large cell carcinomas. And most textbooks use that phrase, large cell carcinomas. These are the poorest differentiated in terms of their state, and they're almost always going to give you the poorest outcome. And because of that, we actually give them another name too. We call them anaplasia. So the mass that's resulting is anaplasia, which really tells you that this is the poorest differentiated state of the malignant neoplasms. So the anaplastic epithelial cells, they tend to grow in sheets or as nests. So you see them clumped together, but you really, really can't tell any characteristics of that other mass that we're going to be dealing with. So here we have normal skin. So here is a couple features that we want to show you. So the cells of the epithelium start down here. This is the basal layer. Right here, all the way down and through. So all this purple that you see. Below it, you will always have a connective tissue. And this makes up what we call the dermis. And those who've taken histology, this is dense irregular collagenous connective tissue. And up in these little papillae regions, this is loose connective tissue, all down and through here. Okay? So the basal layer is here. They're usually columnar-ish or loco or cuboidal-ish in terms of their shape. And then as they go up ladder here, you can see that their shape starts to become more squamous or very flat-like until you get to this layer in here. And then as they progress through, they actually die. These cells die. So this top layer of dead cells, this is what's always been referred to that. This is basically the keratinized layer. And we actually give the full classification, tissue classification, as stratified, because it's multiple layers. Okay? You can see a huge number of layers here. So the word stratified means multiple layers. So we call it stratified squamous based on the shape of that top layer of cells right here. Keratinized epithelium, because of this thick, thick, thick keratinized layer. Now this layer here is actually very, very thick for skin. It's most likely from the ball of your foot or something like that. And so just to let you know, it'll vary in terms of its thickness that you see here. So pretty cool. So this is normal skin. Now we want to look at some of these common characteristics that we look down in here. Okay? So we want to see, does the mass that we're going to look at actually have keratin being produced? Does it have cells that are actually growing as sheets or cords like we have in here? You see them all next to each other. What are the shapes of these cells? Are they going to have some squamous-like cells up top? Are they going to have basically cuboidal-ish type cells down in the basal region? What does it look like? So look at these common characteristics. The mass that we're going to look like is definitely not normal. Okay? You may see keratin being produced in different areas of the actual neoplasm itself. So keep these as characteristics only. They'll show some similarities, but this is going to be very, very dramatically different. So this is squamous cell carcinoma of your lung. You do have a small little alveolus here, another one in here, another one in here, but by and large, this mass has grown very large, and it's pushing the normal parenchyma and stroma to the sides and everything else. So it's just growing and growing and growing. But look at the growth pattern. So look at this area here. So this is a region that we actually have a number of columnar cuboidal-ish type of cells, and then as you're going up the ladder again, you're getting very flat-like. So this is the common characteristics that we're looking for. In addition, do we see production of keratin? Not really up here, but we do see it down here. So this is where we have this large red mass that does have some live cells within it, but a lot of this, if you did a stain for keratin, you would actually see this light up very nicely. So this is all keratin located within this region. Normally, you would see it up in here because this top layer cells would actually turn over and become keratinized. But we're not seeing that. We're seeing big pockets, if you would, of keratin throughout this neoplasm. Look at this one here. You see this? This is all keratin as well. And this is one of those keratin pearls. It's just a bundle of keratin that's very nodular in terms of appearance, and we give it the name keratin pearl. And all of this is evidence that we're dealing with a well-differentiated malignant neoplasm of the lung. Now, what were we using as our key? Basically, it was skin, right? So you actually have characteristics of skin growing within your lung. Now, that is a bizarre concept, and that's what squamous cell carcinoma is all about. Here we have another large, basically this is what we call large cell carcinoma. So it is very, very different from the last image that we just looked at. These are all very large cells, but I can't really find any of those common characteristics. Sure, they're growing in nodules like we have here, and sometimes cords, you can call them. We have long groups of them growing together. Here's another one, N for nodule. But you still know it's lung because you're dealing with an alveolus right here, as well as in here, and they're calling these two. But again, they're being squished together because of the mass of this neoplasm as it grows. And in terms of defining what this is, I don't see those telltale characteristics that we talked about earlier. Hence, we would call this large cell carcinoma. This is a poorly differentiated malignant neoplasm. I can't really say 100% of the lung because I'm not 100% certain. I'm pretty much certain that it's from the lung, but I can't be 100% certain. And that's the problem with some of these malignant neoplasms. You won't be able to tell where they originated from unless you can see definite signs that it originated here. It could be, but it's not 100%. Another type is called the adenocarcinoma. Now, we dealt with the term adeno before. It's glandular of origin. So you're going to expect mucus being produced within some of these areas, and that's fine. And carcinomas, now we're dealing with the malignant version of that. And again, we're talking from the bronchi. Sometimes within the bronchioles, but mainly from the bronchi. And the major feature, we're looking for a glandular pattern. That's what we want to see. In other words, do we see evidence of mucus being produced? And these neoplasms also invoke a local inflammatory response. So you might see lymphocytes present, as well as macrophages. And basically, if we don't have any evidence of mucus being produced or anything like that, then we can group them into the large cell carcinoma category. So what are you looking at here? First things, you can see that, yep, we're in lung. Here's an alveolus, alveolus, and alveolus. But what we're looking at here, this is the mass here, as well as here, here, all of this. Look for the common characteristics that we talked about earlier. Do you see any evidence of mucus being produced? Yes, you do. All of this opaque areas are places where mucus have been produced. All these areas here. So this is growing generally in this direction out this way, and it's going to be spreading and spreading all the way through. So we call this adenocarcinoma of the lung. So it's glandular in terms of its origin because mucus is being produced. We say large amounts. In addition, that local inflammatory response, these are all the immunology cells that are being produced, as well as over in this area here. You see huge numbers, all these dark cells that we see. These are all lymphocytes that have come to this area and that will be battling this specific type of cancer, and this spreads. And basically what happens is some of this mucus can be secreted, and you end up with large amounts, large volumes of mucus within your lungs, and you'll have to have that removed periodically because it does fill back up depending on the size of the mass. So now we're going to get into the biological properties of neoplasms. Now, it's interesting because when we're looking at normal tissue, many normal tissue can actually grow faster than a neoplasm. If you take reindeer antlers, for example, that's one of the best examples. You can actually grow given the season, an inch of antler within a couple of days. It's phenomenal the rate of growth that happens within these antlers. So it's not a rate phenomena. So neoplasia is not a rate phenomena. It is a matter of control. It's how do we get control of those cells? Because right now they're not controllable. They're autonomous in terms of their growth. That's the problem. The other characteristic, if you would, is that many of these neoplasms only have a small portion of these cells that are actually doing the proliferation. In fact, it's more towards, when we're looking at these cells that are growing towards the outer periphery, that are actually doing the growing. And the ones which are usually more differentiated are the ones that are not growing as much or as fast. And we can actually call this, in the more differentiated non-growing ones, and usually more towards the center. And that actually leads to a couple of conditions. We'll talk about that in a minute. So the ratio can actually be called what we call the growth fraction index. And it's how fast this, it really is an index of how fast a specific mass is growing. And with neoplasms, it's usually very small in terms of that, which really indicates that most of this neoplasm is composed of what we call non-proliferating cells. And this actually is a pretty good definition, because when we talk about cancer stem cells later on, you'll see how most of that is only done by the actual cancer stem cell, not by the actual cells that make up the rest of the mass. So more on the biological properties. We talked about carcinomas, and we haven't really talked about sarcomas too much. Well, carcinomas, like we talked about, are malignant neoplasms that are of epithelial origin. In other words, these are the cells that line your tubes and things like that. Sarcomas are malignant neoplasms that form within your connective tissues. So in other words, when you're dealing with fibroblasts and the other types of cells, they can undergo autonomous growth as well. And when you're looking at it, those specific carcinomas and sarcomas actually grow differently because of the nature of those cells. Connective tissue cells tend to, when they proliferate, when they divide, they tend to separate from each other. And then one will go off its merry way, do something, and the other will go off its merry way and do something. That's connective tissue. Epithelial tissues, they tend to grow together. They hold on to each other, and they form occluding junctions with each other and things like that. So they really like each other. They want to grow beside each other. So they tend to grow in sheets and nodules and things like that. So we see those respective normal characteristics within their corresponding neoplasms as well. And that's what we want to look for. So carcinomas and sarcomas, they grow differently. So when we're looking at carcinomas, they tend to stay within the sheets, or they form these solid clusters and nodules. So they have this close cell association. Now the problem here is if you're starting to grow as solid clusters, nodules, and you continue to grow, problem is you're not going to be able to survive very long as a very large mass. Why? Because you've got to be fed. You have to have oxygen. And so you may see that some of these neoplasms undergo what we call ischemic necrosis. Now, you know what this word necrosis means. Basically, it's a pattern of tissue death. Ischemic basically means that we've now lowered the oxygen level within that area. So if you're growing too much, you're not going to have oxygen being able to diffuse through all those cells. So the cells near the center tend to become necrotic. And then you see them as basically as a pattern of tissue death. And we call that ischemic necrosis. Now, why is this happening? Well, because oxygen can only diffuse so far. And as a result, you can only grow so big. As a result, you'll see long cords of these cells rather than these big, huge nodules. In order to get a blood supply, you have to secrete angiogenesis factors. So that's key there. And once that happens, you'll get what we call tumor vascularization, which I don't like the word tumor, but nevertheless, that's what the general term is. And then all bets are off. You now have a supply of blood that you can receive nutrients, oxygen, and potentially that will be a traffic area where you can actually move around. So that's with the carcinomas. Within sarcomas, because they show the separation once the cells divide, then they're less likely to have ischemic necrosis. Why? Because connective tissue by its inherent nature has a number of blood vessels found within it. So if you're a mass that's growing, these tend to be quite large because you've got McDonald's basically right next door, right there. You have the ability to have vasculature within the mass, and that's because they're there already. You don't need angiogenesis factors, although that does happen. You get more vascularization that's going to be taking place. So you've got your food supply and you have your oxygen, and you can take away the nasty waste product, carbon dioxide. So ischemic necrosis is less common in sarcomas because of that. And just by physically feeling them, sarcomas tend to be very soft and fleshy, while the carcinomas tend to be much, much harder. You can actually feel the outlines of some of these masses. So how do you get this? What's the nature of, or the biological nature of, basically cancer development? And one of them, probably one of the best models that I like, at least in terms of chemical carcinogenesis, is a three-prong motion. Three-prong, 3 steps that will essentially give you ultimately a neoplasm down the road. And we're talking very, very long. This is a chronic disease. As a result, you would probably see from initiation right down through full-fledged neoplastic formation, it could span the gauntlet in terms of 10, 20, 30 plus years in order to get that. So we're talking very, very chronic. So the model we're going to talk about basically has 3 steps, what we call initiation, promotion, and progression. Now I highly recommend you read that article by Emmy Farber and Dr. Sarma. It is really an interesting read. It goes through chronic disease, it goes through disease processes, and it's really a very enlightening argument that they present. Now, the model that they do present, based on what we call the resistant hepatocyte model, is still around today. And basically, when you develop a conceptual model, which is what they did, then the model leaves room for adding more features to it. So basically what genes are involved, other changes, whether it's mutations or epigenetics, etc., etc. Now, in terms of the initial stages, so we're up here in what we call cancer initiation. We have our individual cell, and we have some sort of mutation. That's fine. I'm going to stick with this diagram here. I left this one. This is one of the older diagrams that we would use. And one of the reasons why we don't use it as much is this one's a bit more colorful, and it has another change that I want to highlight to you. And we'll talk about that briefly in a minute. So we have some sort of basically what we call a biochemical change that happens with your DNA. And so by and large, it's going to be a mutation. So step number 1 in terms of cancer initiation is a biochemical change on your DNA. And this is fixed in that DNA when you undergo 1 round of cell proliferation. Now, if you actually have a very elaborate DNA repair system, if that is repaired within about 96 hours, then that cell will go back to normal. But if that repair is not done and that mutation holds, usually the cell will replicate once, and now that damage is now fixed within that cell. That biochemical lesion, the nature of that, is usually by the chemical carcinogen. And that, by definition, is initiation. We now have an initiated cell. And this undergoes further development through what we call promotion. where we start to see more and more genes being affected. And this can be considered in terms of what we call getting promoted to the next level. And details you can actually go within that specific article. It's actually quite neat from that perspective. And there are actually certain chemicals that can help promote this initiated cell to the next level. And that next level will be getting into more down the pathway to a resistant hepatocyte. So as we get into progression actually takes the longest stages. And here we have more fundamental changes that are happening within the cell. We have more mutations that are happening. We can have epigenetic changes that will favor growth of the actual cell. And as the cell grows, it's going to be almost like down in this area here where we're going to actually start to proliferate and select. This is where we start to get what we call the resistant hepatocyte. We're getting clones, if you would, of these cells. And they're actually now forming nodules and cords. But they're still not cancer at this point. So they're growing and growing, but they're still not cancerous at this time. What we want to essentially happen as we get down to these stages here is more genes being turned on that promote growth and autonomy, and we have more via the epigenetic changes as well. And it's just too many to really go into. But somewhere along this particular timeline, we now have a change within the cell that actually seals the fate of the cell in terms of autonomous growth. So once it gets into that area here, then basically via clonal expansion, this is going to cause this mass to increase in size. And it's all because if it's a single cell, this is where we get essentially that truly resistant hepatocyte, if we're dealing with lung hepatocellular, what we call hepatocellular carcinoma. And up to this stage here, most of these nodules that are forming don't go anywhere. They actually don't. They just stay. But there's always that one who undergoes subsequent changes that favor the growth and the autonomous growth of that specific resistant hepatocyte. And that's why we call it a resistant hepatocyte, especially at this point here, because they are no longer responding to our environmental perturbations. In other words, we send signals out for them to stop growing, but they don't obey those signals. They continually grow. So now we have an expansion of this resistant hepatocyte into multiple cells. And this is really where that neoplasm really starts to grow and grow and grow. And these steps, like I said, this all takes a very, very long period of time for that to happen. This is just showing you a different perspective. So we now have our initiated cell. So in other words, we had a biochemical change followed by one round of cell proliferation to fix that DNA damage. And then we start to see growth happening. Okay? But at this stage of the game, it's a response only. It's not necessarily cancer. Okay? So generally, sometimes we'll call it metaplasia. Other times we'll have hyperplasia, where we just have an increase in cell number. And then, as we get to what we call dysplasia. So dysplasia we really haven't taken, but we've shown you the definition before. It's an alteration in the size, shape, and organization of these mature cells. But we're still not neoplastic at this point. So this is nothing, not cancer, but it's on the road to. And it's usually a pretty good diagnostic tool that we can use to say, hey, this could develop into neoplasia down the road. So we do use that in certain circumstances. When we get to what is called in situ cancer, I don't like that term. In situ carcinoma is a better term, depending on the location. This is neoplasia. So now the fate is this area here, that one of these cells basically went along and became neoplastic. So now it grows autonomously. And so one of the first stages that we see is in situ. Basically, it stays there. It doesn't invade along within the basement membrane. The basement membrane is still intact. So it's what we call right there. It's in situ. It's sitting there. And hence the word in situ carcinoma. If this mass produces specific cytokines and growth factors that favors vascularization, in other words, angiogenesis factors are being secreted, then we can actually have blood vessels grow out into the mass, and then, wow, you have now a scenario where you can feed the mass, you can actually give it oxygen, take away waste products, and it's an actual vector that you can actually transport some of these cells to distant sites. So cancer metastasis can actually take place. This is full-blown invasive cancer. And almost always, this will lead to spreading and ultimately the poorest prognosis. And within this mass, you can actually look at those characteristics we talked about earlier and determine whether or not you have a well-differentiated malignant neoplasm or a poorly differentiated malignant neoplasm. I would probably say that it's more on the spectrum towards the poor differentiated malignant neoplasm, because those masses almost always have the poorest outcomes. So you have to think of it as if you have well-differentiated one end, you have poorly differentiated mass on the other end, that it is a spectrum, okay? And you can actually see different variations of that depending on the nature of that specific mass. Now, that's the problem, okay? And this is what confuses students the most, because they want to see this and this. Well, in terms of neoplasia, you're not going to see that. It's just not going to happen. So what are some of these characteristics that allow these cancers to do what they do? Well, let's take them from the top. We have sufficient growth signals. You want to evade our body's responses, specifically evading apoptosis. You want to basically deny the anti-growth signals. You want to become autonomous in terms of your growth. So that's the way you do that. You want to invade local tissue as well as go to distant sites. You need to do that via the angiogenesis factors. This way you get food, oxygen, and you get your waste products taken away. And this one really is the one. You want limitless replication potential. Let's have a look at each of and give you examples of each of these. So in terms of the self-sufficient growth, you want to activate the oncogenes. And these are examples of the mechanism. There are many, 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 but oncogenes is one. You want to basically become insensitive to the anti-growth signals. So you want to basically stop the tumor suppressors. Evade apoptosis. So you want to basically produce factors that give you survival characteristics. And we'll show you a little of that in a minute. You want limitless replication potential. That's really it right there. Turn on telomerase. Does anybody know what telomerase is? Anybody? It really allows your cells basically to grow and grow and grow. How does it do this? Well, you have telomeres on the ends of your chromosomes. And basically, those are pieces of DNA, and as you age, those get shorter and shorter and shorter. If we turn on telomerase, telomerase adds onto that telomere. And the more it adds on, the longer your cells are going to be living, etc., etc. So if you want limitless life in terms of this, what we call growth potential, turn on telomerase. It's an enzyme that basically is, on most of our cells it should be off, but it's not. And if we turn it on, then you actually set the stage for it to have these telomeres become long in terms of their effect, and they will allow you to replicate basically forever and ever and ever. And one example, or cancer cell lines. Look at the HeLa cells. This is an immortal cell line. And this is because telomerase, at least one reason, is the telomerase is very active within these cells. And so you're allowed to grow and grow and grow and grow. Another one is sustained angiogenesis. You got to feed it, bring in oxygen, take waste products. So you want to basically produce factors that are involved with angiogenesis. And tissue invasion, and you want to move around, basically inactivate E cadherins. These are the integrins, and basically you want to make sure that these cells do not stick to any place. You want to have them the ability to move around, and that's what inactivating the E cadherins will do for you. And the degree of these will essentially dictate how cancerous you are from that perspective. So the more, especially the one that you see quite often, is in fact this one here, limitless replication potential. Please do not memorize the slide. This is here just to show you the complexity of the cancer signaling. But I do want to point out to you certain locations. We have changes in gene expression, and we have multiple pathways that will lead to that. In addition, you have basically changes in cell proliferation or dealing with your cell cycle. And one of the famous ones that everybody talks about is P53. This is your DNA damage sensor. You mutate that, you're going to get cancer. You want cell death as another aspect. You want to prevent that from happening. Caspases tend to be involved with cell death or apoptosis. So if you stop the caspases, then you can actually put a control on that. One of the major molecules that does that is BCL2. And one of the other ones that deals with gene expression, there's so many that deal with gene expression. That's how important it is in terms of the growth factors, hormones, etc., etc. One of the famous pathways is the Wnt beta-catenin pathways. And you'll see this will be mutated, especially in colon cancer at the APC level. Very interesting stories about that. And I encourage you to essentially read further up on these because the stories that they go through are very, very interesting. But the point is, changes in gene expression, cell proliferation, dealing with the cell cycle itself, especially dealing with your ability to counteract any mutations, and that's done by P53. Avoiding cell death, in other words, evading the apoptotic factors that are involved, usually the caspases. You can do that by generating certain other factors that will stop that specific production. Very interesting. So this leads us into what are called cancer stem cells. And I guess the question is what, and this is what confuses students the most, what is a cancer stem cell? And really, the only way you can define that, and we'll give you a sort of a lengthy definition, and then we'll try and distill it down to a smaller definition, is you have to be dealing with a neoplasm. And it's a cell that's found within a neoplasm that possesses the capacity for self-renewal. Not only that, but it has the ability to cause the heterogeneous lineages found within these cancer cells. So what does that mean? Well, when we're looking at a normal stem cell, and I'm assuming everybody understands what a normal stem cell is. So what criteria, think about what criteria you need in order to be a stem cell. One of them is self-renewal. That's by definition part of what a stem cell is. The other part is you have basically the potential to differentiate into various lineages. So that's the other part of a stem cell. If you're a cancer stem cell, you have those specific features inside of a neoplasm. And that, by definition, it's an operational definition, that by definition is a cancer stem cell. And I like the definition of, this specific definition of what a stem cell is. And this was done back in 2013. And the author is Biaconi. And when we're looking at normal stem cells, there's two essential biological functions of a normal stem cell. And it's so true. They hold and protect our DNA from zygote to death. And they divide to originate the huge, huge number of cells that make up our body. You know, that's a philosophical definition of what a stem cell is. So these cells are extremely powerful in what they can do. Not only can they self-renew, so they have an identical copy of each other, so they undergo, in reality, stem cells undergo both symmetric as well as asymmetric division. Asymmetric division really is you're making two daughter cells. One's a normal stem cell, that's your self-renewal, and the other is a cell that will be differentiated into something. And so that's the asymmetric division. The symmetric division is basically you're creating more stem cells of yourself. And this is the self-renewal process. And it's really, really complicated from that perspective, but nevertheless, that's the gist of that. So a cancer stem cell, you have to be looking at a neoplasm in order for it to be a cancer stem cell. And it has those characteristics of stem cells. Self-renewing ability, and it's basically all the other cells that make up what we call the heterogeneity of the tumor is also done by the cancer stem cell. So when it divides, you're getting a cancer stem cell being shunt off, as well as a normal, what we call cancer cell, essentially, that's going to be part of that mass. And usually the heterogeneity of that comes out as they start to divide again and again and again too. So they can all divide within certain reason. And then you'll see how these cells down here will differentiate into something different. So that's the idea behind cancer stem cells. A little bit more. Now, when we're looking at cancer stem cells in terms of its self-renewal, we really thought that cancer stem cells, like they have here, when they're dividing, you would give off another cancer stem cell, and you give off another type of cell that differentiates into something. Now, there's another model that's out there, and it has to do with what we call cancer stem cell plasticity. And this is the scary part, because for the first time, they're actually saying that a cell that came out of a cancer stem cell, right here, that normally differentiates into something else, can actually revert back into a cancer stem cell. So if the tumor actually has only a certain number of cancer stem cells, once they're gone, that's the end of the tumor. Basically, it'll be slow growing, etc., etc. But now we have a scenario where any one of those cells can actually revert back to the cancer stem cell. So we have the self-renewing process will continue and continue and continue. That's a pretty scary thought. Not only that, but we have both intrinsic as well as extrinsic, which is the tumor microenvironment, influences on these cancer stem cells. Now, I highly recommend that you have a look at that paper that we have posted on Quercus. It goes to this way more in depth, and it's a really interesting theory. And if this is true, which it probably is, then we're going to have to find better strategies to deal with these cancer stem cells. And we'll talk a little bit more about that later on. But what I want to talk about is now what we call the tumor microenvironment, or the cancer stem cell niche. The niche is really the action place. This is where the cancer stem cells will be influenced by outside sources. So this is really extrinsic factors that are coming into play. And this includes a whole host of different types of environments that the tumor can actually thrive in. One of them, hypoxic conditions, would really leads to cancer metabolism issues, enhancements, and things like that. Basically, the influence of connective tissue within that environment, the extracellular matrix, all these fibers being laid down, not only laminin but collagen, etc., will have an effect vascular beds, in other words, tumor angiogenesis factors. You want to basically get that tumor fed, oxygen delivery, and CO2 taken away. So this is another external niche that will enhance the malignant neoplasm. And another interesting one is what we call inflammation. This is really interesting. This is an active new area of investigation. Let me show you. So in this particular area, we have what we call macrophages coming into the scene. We call them, and we give them a specific name. We call them TAM, tumor-associated macrophages. What? Well, it turns out that macrophages within this area can actually enhance certain neoplasms. Specifically, they secrete factors, all of these here, that have basically a positive influence on the tumor growth or neoplasm growth. It also plays in with multiple niches. So here we have a scenario where the extracellular matrix can be remodeled by these macrophages to, again, in favor of the neoplasm growth. So we're talking getting rid of essentially the barriers, if you would, getting rid of basement membranes, getting rid of traps and things like that that would normally hold the mass in place, but now we want to essentially let the mass grow and move. So the potential there is for metastasis to take place. Another role is within immune suppression. So that makes perfect sense. If you want the mass to grow, you want to eliminate your immune system so it doesn't react to it. So these macrophages, these TAMs, actually secrete cytokines that have a negative influence on your immune system. We call this immunosuppression. So this includes T helper cell inhibition. We want to get rid of as much of this as we possibly can. Stop the B cells from functioning, etc., etc. In addition, angiogenesis. We've talked about this multiple times. We want to be able to feed and give blood supply to the actual neoplasm itself. And we do that by secreting angiogenesis factors within the area. And this allows for tumor vascularization. And this is all done by these tumor-associated macrophages. And this is a really interesting new area of research that is going on. So, do cancer stem cells really exist? Well, yes, they do. We have evidence to show that they do exist. So, for example, on these cancer stem cells, you can actually have a whole series of markers that we can use to identify these cancer stem cells. One of them is basically what we call CD44 positive, CD24 negative, and lineage negative. Basically, this type of cancer stem cell, which is on the cell itself, is a code that tells it that, yeah, we're dealing with a cancer stem cell. And the evidence for that is if we take these cells and inject them into a mouse, like we have here, the CD24 negative cells grow a very large mass, a neoplasm, versus the cells that are CD24 positive do not grow a mass at all. And this is the corresponding histology. This is the normal histology. And this here shows you the actual basically neoplasm. It's a, for all intents and purposes, a malignant mass that's grown. So this is evidence that, yes, cancer stem cells do exist. Strategies to deal with them. This is something that we haven't really done a lot of. It's really in its infancy at this point. So what we want to do, most of the time we're dealing with, if we have a tumor, we have a mature tumor, and we basically have drugs that will deal with the actual tumor itself. That's not good enough nowadays. Why? Because if we don't get it all, hopefully most of the time we do get it all, then they can come back. And that's the whole problem with cancer stem cells, is it's a very small population that has this self-renewing ability. So when we do a global type of treatment, then you may miss out on getting these cancer stem cells. That's the problem. If you do miss out on these cancer stem cells, basically the mass will come back, the neoplasm will come back. So we need specific treatments that deals with getting rid of these cancer stem cells as much as we possibly can. We want to take these cancer stem cells and drive them down to apoptosis. The more we can get rid of, the less likely we'll get a large tumor. And that's the whole point. We want to get these cancer stem cells, get rid of them, and the resulting neoplasm will hopefully be much, much smaller and respond better to other types of chemotherapy.